Eukaryotic protein synthesis. The central dogma of molecular biology states that DNA codes for RNA, which codes for proteins. DNA is the molecule of heredity that passes from parents to offspring. Protein synthesis occurs in cells. Transcription occurs in the cell's nucleus, and translation occurs in the cytoplasm. This is my model for DNA. In eukaryotic cells, DNA is found in the nucleus of the cell inside of the chromosomes. Although my model does not show this, the shape of the DNA is the double helix. It is made of deoxyribonucleotides. The backbone is made up of deoxyribose sugars and phosphate groups, held together by phosphodiester bonds. There are 4 types of bases in DNA: adenine, guanine, cytosine, and thymine. Adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds. Adenine and guanine are purines, and thymine and cytosines are pyramides. The 2 strands of DNA are antiparallel. One strand goes from 5' to 3', and the other goes from 3' to 5'. The purpose of DNA is to code for proteins, which are responsible for traits. This is my model for messenger RNA. mRNA is found in the nucleus during transcription and then moves out of the nucleus into the cytoplasm. mRNA is single-stranded and varies in length from hundreds to thousands of nucleotides long. The 4 types of bases in mRNA are adenine, guanine, cytosine, and uracil. Messenger RNA is made of ribonucleotides. The backbone is made of ribose sugars and phosphate groups, held together with phosphodiester bonds. The purpose of messenger RNA is to be the template for translation. It carries genetic information copied from DNA to ribosomes in order to make proteins. This is my model for transfer RNA. tRNA is found in the cell's cytoplasm. It is 70 to 90 nucleotides long, and although my model doesn't show this, it has the shape of a clover leaf. Its bases are A, U, G, and C, with 2 hydrogen bonds between A and U and 3 between G and C. The backbone of tRNA is the same as mRNA. The purpose of transfer RNA is to deliver the correct amino acids to the polypeptide binding sites. Each tRNA has an anticodon, which is the complement of the mRNA codon. tRNAs move through the 3 binding sites in the ribosome from A to P to E, except for when the start codon is detected, which moves from P to E. This is my ribosome model. The ribosome is made up of a large ribosomal subunit, a small ribosomal subunit, and has 3 polypeptide binding sites. It is made up of rRNA and proteins and is found in the cell's cytoplasm, as well as the rough endoplasmic reticulum. The purpose of ribosomes is to convert genetic code into amino acid sequences and build proteins. Transcription. The first step of transcription is initiation, which happens in the cell's nucleus. It starts at the promoter region, which is also known as the Tata box, a section of DNA where there is plenty of nucleotides with bases A and T, which are easier to break through because they are held together by 2 hydrogen bonds rather than 3. Promoter region allows the enzyme RNA polymerase to bind and open the double helix by breaking the hydrogen bonds and exposing the template strand. The promoter region does not get transcribed into mRNA. The next step of transcription is elongation. During elongation, RNA polymerase reads the template strand on DNA in the 3' to 5' direction, pairing complementary bases to the template strand and building the mRNA strand in the 5' to 3' direction. As RNA polymerase goes along, the mRNA falls off and the DNA reanneals behind it. Building blocks are A, U, G, and C, which come from the cell's cytoplasm from older messenger RNA that has been hydrolyzed. Elongation happens very fast, attaching 40 nucleotides per second, and ends when RNA polymerase reaches the terminator sequence. Next is termination. At the end of a gene, there is a terminator sequence that signals the RNA polymerase to stop transcribing. The mRNA strand and polymerase are then released and the DNA remains unchanged. The terminator sequence is a string of adenines in eukaryotes and a hairpin loop in prokaryotes. Post-transcriptional modifications are also a part of transcription. One modification is the addition of a 5' cap on the messenger RNA, which is made up of 7 guanine nucleotides. The function of the cap is to be recognized by the ribosomes and to protect the mRNA from digestion in the cytoplasm. Another modification is the addition of a poly A tail on the 3' end of the mRNA. It is made of 50 to 250 adenine nucleotides. The function of the poly A tail is to help with export from the nucleus and keep the messenger RNA from being degraded. The last modification not shown in my model is splicing. During splicing, the intron sequences, which are non-coding, are removed from the mRNA strand, leaving the exons. Splicing joins the coding sequences together. The overall purpose of post-transcriptional modifications is to allow the mRNA to be translated, as well as to protect the strand from being degraded in the cytoplasm. mRNA can now exit the nucleus. Translation. The first step of translation is initiation. Its assembly is the attachment of the small and large ribosomal subunits and tRNA on the messenger RNA. The ribosome uses a reading frame, a system used to separate the base pair sequence into readable codons. The ribosome moves along the mRNA until it reaches the start codon, AUG. The start codon is detected, the first tRNA with the anticodon UAC attaches to the P site, delivering the amino acid methionine in order to begin the polypeptide chain. The next step is elongation. The ribosome has 3 binding sites, the exit site E, the peptidyl site P, and the amino acyl site A. The binding sites accommodate the transfer RNAs that enter the ribosome at the A site. Anticodons are found at the base of tRNA and bind to the corresponding codons on the mRNA strand. The table of codons is used to find which amino acid corresponds to each codon. Each tRNA carries one specific amino acid, and some amino acids have more than one possible codon. The wobble hypothesis allows some transfer RNAs to recognize more than one codon. At the exit site, the tRNA exits, leaving an amino acid which binds to the next amino acid with a peptide bond. This process continues, creating a long chain of amino acids until a stop codon is reached. Termination comes next. During termination, a stop codon, UGA, UAG, or UAA, is reached on the mRNA strand and signals protein synthesis to stop. A protein release factor is introduced, and the polypeptide chain, ribosomal subunits, protein release factor, and mRNA strand are all separated, ending termination. The polypeptide chain now needs to be made into a protein. A protein is a string of amino acids that is taken on its shape through processing and folding. Proteins can have primary, secondary, tertiary, or quaternary structure. Primary structure is a sequence of a chain of amino acids. Secondary structure is local folding of the polypeptide chains into helices or sheets. Tertiary structure is three-dimensional folding pattern of protein due to side chain interactions, and quaternary structure is protein consisting of more than one amino acid chain. Sometimes the proteins need to be processed, and a few amino acids will be clipped off in order to make the protein fully functional. If a mutation occurred in this process, it could have an effect on the organism. Substitution mutations usually don't cause a large effect. However, insertion and deletion mutations may cause a frame shift, which could lead to more harmful effects, such as sickle cell anemia or cystic fibrosis. Thanks for watching!